I think some thoughts on therapeutic approaches. So we actually are very fortunate to have a number of these new antibody-drug conjugates in development. And I'd like to get your thoughts basically on some of these, in addition to anti-CD30, that might bring these agents to the armamentarium of therapeutic choices for our patients. First, before we go on, we know that CD30, but also in addition to CD30, there are two CD19 antibody-drug conjugates, one from Immunogen and one from Seattle Genetics. There's actually a CD22 and a CD79B drug conjugate. The two of those actually, and it's very similar, the payload is often antitubular toxins, and so they're very similar agents, but the difference is when you enter the cell between a CD22 and a CD79B, even though you're using the same target or the same drug, is there potential differences in how they're actually being processed, delivered to the tumor cell, et cetera. So there's very interesting things that we still have to learn. And one other CD22 that was actually published from Pfizer was the inotuzumab ozogamicin. A number of the people on the panel here have used it. We actually were part of their phase 2 studies and other studies. And of interesting, there was actually a clinical trial with that between using that in patients with relapsed refractory large cell lymphoma and then randomizing that to basically dealer's choice. And it actually had to be stopped early because of futility. It didn't show that there was enough activity with a single agent monotherapy in that kind of patient population. But I think it's interesting that these agents, and what I think is another important thing, is that the reason CD20 isn't used as an immunodrug conjugate is that these are actual antibodies, 30, but also 19, 22, and 79B, that when an antibody binds to its target on the tumor cell, it internalizes. So we have to use the biology of these actual interactions to the best of their abilities. That makes a lot of sense that you would add something. Without the linker technology, we don't have it. Now with the linker technology, we see what benefit we have with very toxic therapies. But people in the past have used bacterial toxins, which unfortunately may be too immunogenic. The reason that they weren't successful early on is that the human immune system says, what's this bacteria doing here, and it goes and attacks it. So I think we've been very lucky. We don't see a lot of immunogenicity with these agents. But I think it's very interesting because emerging, and even at this year's ASH, is a number of these are being presented either in poster or oral presentations in B-cell lymphomas, because these are actually B-cell targets. And I was just wondering if you have any comments from the group. We know I think it's kind of early. I don't think there's going to be a lot of discussion here. But I think it's interesting that we are seeing signals with respect to B-cell neoplasms. Interestingly, maybe it's not going to work the best in patients with bulky disease, because you have to be able to deliver a drug to too many sites, too many cells. But with data from other studies, it suggests perhaps having patients debulked or have less tumor, this may be the best way of using these agents. Anybody want to comment? I mean, there's been data in refractory relapsed diffuse large B-cell lymphoma with the 19, and also was mentioned with the Genentech Roche 22 and 79B. The early data is there, but what more do we need other than seeing response rates in the 30 to 40% range? What more do we need? We'll ask on us.
Right. So I think we need to look at these agents now as a class. So it is going to be interesting to watch how these agents evolve. I think the success of brentuximab vedotin and then HER2-targeted agent breast cancer opened the door wide open for these agents by delivering really chemotherapy-like agents, but in a more targeted way that would allow you to deliver higher doses to the tumor cells compared to other targets. So I don't think it would be realistic to expect similar responses that what we've seen with anaplastic large cell lymphoma and Hodgkin lymphoma. But I think some of these responses may be sufficient to move these agents in combination as long as there is no overlapping toxicities with other active agents. It could be combined with biologics or chemotherapy, depends. But I think there is room for combinations in the future, even though the response rate as single agent is lower than what we've seen with brentuximab.
Any other comments?
Well, looking at the abstracts from this year, some of these drugs have been in combination with rituximab, where you wouldn't think there would be additive toxicity. And interestingly, in some cases, the rituximab is beneficial and in some cases not. So that's going to have to be worked out for each one of these drugs, what the appropriate combination is.